we are continuing our study of the molecules of life, organic macromolecules. Specifically today, we're going to study proteins, and this material is covered in the Campbell Biology in Focus textbook, Concept 3.5, on pages 55 to 63. So make sure you have read those pages before you begin this lesson. A protein is a polymer, and the basic building block that makes up that polymer is called the amino acid. So when we were discussing carbohydrates, we said that a polysaccharide, like starch, is a polymer of glucose. In other words, starch was a long chain of glucose bonded to glucose, bonded to glucose, bonded to glucose. Each glucose was like a pearl. The entire starch molecule was the pearl necklace. We have the same pattern here. Each amino acid is one individual pearl, and if you join hundreds or thousands of amino acids together, you build a long, complex chain called protein. So that's what we mean when we say that a protein is a polymer composed of many amino acids. Most of us are familiar with foods that are rich sources of protein, such as meat, eggs, fish, beans. Soybeans in particular are really high sources of protein, and dairy. So if a protein is a polymer made up of individual amino acids, then it's important for us to understand the structure of an amino acid. So this is the general structure. All amino acids have this same basic structure. So every single amino acid has a nitrogen. Notice we're not just talking about carbon and hydrogen and oxygen like with most lipids and all carbohydrates. Now we've got nitrogen. So it's a nitrogen atom bonded to two hydrogen atoms. That makes up what we call the amino group. And on the other side of the amino acid, we have a carbon that's double covalent bonded to an oxygen, and then single bonded to another oxygen, which is also bonded to a hydrogen. So this little area is called the carboxyl group. This carbon is known as the central carbon, and it is always bonded to a hydrogen. And then we have the most interesting part of the amino acid, the R group. Sometimes it's called the R side chain. And what you need to realize is that there are 20 different things that can be plugged in for R. There's 20 different atoms or molecules that can be stuck in that spot where you see the R. Thus, there are 20 different amino acids. And I'll show you a picture of what some of those amino acids look like. In fact, I think I have all 20 of them represented in these two slides. So the simplest of all amino acids is called glycine. Here's the amino group. Here's the carboxyl group. Notice that that part looks exactly the same for every single amino acid. Amino group, carboxyl group. Amino group, carboxyl group. The only thing that makes a glycine different from an alanine, the only thing that makes an alanine different from a tryptophan is what you plug in for R. And the R group could be as simple as just a plain hydrogen in the amino acid glycine, or a complex ring structure on the amino acid tryptophan. Some amino acids are nonpolar, meaning the R group does not have any electrical charge. And if it's nonpolar, that means it's not going to be attracted to water. It's going to be hydrophobic. Other amino acids are polar, which means that somewhere on the R group, there's going to be some sort of at least partial electrical charge. These polar amino acids are going to be attracted to water. Some amino acids act like acids. They have acidic R groups, and some have basic R groups. So they act like a base. So it's a pretty diverse group, but you can tell from these pictures that what makes an amino acid, electrically charged or not, polar or nonpolar, is the R group. It's the R group because the rest of the amino acid is absolutely identical. So again, there are 20 different naturally occurring amino acids which are used to build proteins inside living cells, bacteria, plants, animals, fungus, etc. Now, whenever you have a monomer, such as an amino acid, and you want to join it together with another monomer, another amino acid, you simply do dehydration synthesis. We've talked about this before. Dehydration synthesis is used to join monosaccharides together to build disaccharides. It's used to join fatty acids onto glycerol to build a fat molecule. And once again, we're going to have an enzyme that comes in and removes an oxygen and a hydrogen from this carboxyl group, and we're going to remove a hydrogen from this amino group. So if you take those three atoms, you pull them off, that makes a water. Dehydration, removal of water. And then we can take this carbon and bond it directly onto this nitrogen. Boom, right there. See that bond? That's a brand new covalent bond. It even has a special name. It's called a peptide bond. A covalent bond joining together two amino acids is called a peptide bond. And this molecule is no longer a single amino acid. It's two amino acids bonded together by a peptide bond. We call that a dipeptide. Dipeptide is two amino acids bonded together. Now, if we were to add a third amino acid, and a fourth, and a fifth, and a sixth, we could keep adding on to this and build a very long chain of amino acids. We call that a polypeptide. So all proteins are polypeptides, or they're made from polypeptides. You can't build a protein without building a polypeptide because that's basically what a protein is. It's a long chain of amino acids that have been joined together like pearls on a necklace by the process of dehydration synthesis. Okay, so after amino acids have been assembled into a finished protein, this is done inside cells by ribosomes. And after the protein has been built, there are 4 different levels of structure, 4 different levels that we can use to describe the structure of a protein. So let's go through those 4 levels quickly. This can be a little bit confusing. First of all, every protein has what's called a primary structure. So I'm looking at this protein, and notice it is like a pearl necklace. Here's a pearl, here's a pearl, here's another one. This is a long chain of pearls that have been assembled into one long necklace. Well, of course, each pearl is an amino acid. So you see this LYS? I don't know if you can read that. That stands for lysine. That's the name of an amino acid. And it is bonded to valine, which is bonded to phenylalanine, which is bonded to glycine, arginine, cysteine, and so forth. You don't need to know the names of all those amino acids. But if I was to ask you what is the primary structure of this protein, you would simply read out the names of the amino acids in order. This particular protein has a lysine bonded to a valine, bonded to a phenylalanine, bonded to a glycine, and I could keep going. I won't, I promise. That'll put you to sleep. But if I was to call out the names of all these, all the way to the final leucine at the end, I would have just described the primary structure of that protein. But proteins don't just have primary structure, because remember, each one of these amino acids has a different R group. Remember the R group? Let's go back. Oh, we can look at it right here. There's the R group. And some R groups are electrically charged. Some are not. Some R groups are hydrophilic. They're attracted to water. Some are hydrophobic. They turn away from water. So, excuse me. So what happens sometimes is you might have, and I'm just going to make something up here. This is probably not even accurate, but just for a simple example, this amino acid might have an electrically charged R group, and maybe this amino acid right here also has an electrically charged R group. So maybe this one has a slight positive charge and this one has a slight negative charge. So those two amino acids, which are pretty far apart, I mean, look at how many amino acids are in between them, probably 10 or 12. They're pretty far apart, but these two might be attracted to each other because of their electrical charge. And so even though they're far apart, they want to get as close together as they can. And so what happens is, because of that attraction between different amino acids along the chain, the entire protein or portions of the protein will sometimes twist or pleat. And I've got a picture that shows that on the next slide. So here would be what's called a protein helix, an alpha helix, which is just a protein that has twisted around and kind of coiled up. Or the protein will bend back and forth to make a pleat. This is called a pleated sheet. And this happens simply because the R group of one amino acid is hydrogen bonding with the R group of another amino acid further down the line. We call this secondary structure. It's the bending or twisting of a protein due to hydrogen bonding between amino acids. And this is a really good picture because not only does it show the secondary structure, which you can see down here, but if you look in the larger zoomed out picture, we can see that the entire protein is folded. So portions of it are twisted, portions of it are pleated, but the entire protein has been folded into a complex three-dimensional shape. And that three-dimensional shape is called the tertiary structure of a protein. This refers to the folding of a protein into a three-dimensional shape. And this three-dimensional shape is partially maintained by hydrogen bonds. So this would be an R group that's located on this amino acid. Here's an R group that's on this amino acid. And if those two R groups are attracted to each other, they're going to form a hydrogen bond. It's going to hold that folded protein in place. But sometimes there's even stronger bonds, like a disulfide bridge, which is a sulfur atom covalently bonded to another sulfur. Some R groups contain sulfur. Or maybe even an ionic bond. And there are other types of bonds that can hold this folded three-dimensional protein into its shape. Now you're going to find the more we learn about proteins, the more you're going to realize that every protein has a complex three-dimensional folded up shape. Again, that's called tertiary structure. And the shape is everything. If a protein loses its three-dimensional shape, or if the three-dimensional shape changes in any major way, that protein no longer functions. So it's very important that these hydrogen bonds and these covalent bonds and these ionic bonds hold that folded protein in exactly the right shape. Now, for many proteins, this is as complex as it gets. I would say most proteins have 3 levels of structure: primary, secondary, and tertiary. Remember, primary structure is just which amino acids are on the protein and what order are they in. Secondary structure is where you have small areas of twisting or pleating because of hydrogen bonds in between the R groups. And then finally, tertiary structure is the complex, folded up, three-dimensional shape that proteins typically take on. There is a 4th level of structure, but not all proteins have the 4th level. A good example of a protein that does have the 4th level is hemoglobin. So here's a good picture of a hemoglobin protein. And you notice that the hemoglobin protein is so large and so complex that it's actually made up of 1, 2, 3, 4 separate smaller proteins. It's one big protein that's made up of multiple polypeptides. So this, where it says beta chain, this is one polypeptide that was assembled. It has folded up into its tertiary three-dimensional shape. Here's another polypeptide that has also folded up into its tertiary shape. A third and even a 4th polypeptide. And they've all been formed and they've all been folded. And now those 4 polypeptides are snapped together to form this huge functional protein called hemoglobin. So when you have a single protein that's made up of multiple three-dimensional polypeptides, we say that protein has not just primary, secondary, and tertiary, but it also has quaternary structure. This refers to the final three-dimensional configuration of a protein, a really large protein like this, that is composed of 2 or more, and see, it can be 4 in the case of hemoglobin, separate polypeptides that are sort of pieced together like the pieces of a jigsaw puzzle to make the one functional protein. Okay, so now that you understand the structure of a protein, let's go through some examples. There are literally hundreds of thousands of proteins that human cells make. And so obviously we cannot learn them all, but I'm going to give you a sampling of some of the more common and more important proteins. And I want to make sure that you know the function of each. Collagen is a strong, stretchy protein found in ligaments, tendons, and cartilage. So cartilage would be like your external ear, the part that's kind of bendy. or your nose, the part that wiggles back and forth underneath the skin of your nose, that's cartilage. Those things are all made out of this very strong elastic protein. Keratin is a strong structural protein found in skin, hair, nails and claws, scales, think the scales of a reptile or a fish, and also horns, like the horn of a rhinoceros. Actin and myosin are special proteins inside our muscle cells that allow for movement. They allow muscle cells to contract and relax, to shorten and lengthen. So movement is made possible by these very special, unique actin and myosin proteins. Hemoglobin is the protein that makes our blood red. It is a protein found in red blood cells. Remember, we just talked about it because hemoglobin is an example of a protein that has quaternary structure. And the purpose of it is to transport oxygen inside our red blood cells. Here's some nice photos. In this close-up of a hair follicle, you can see the layers of keratin protein on the surface of that hair. We can see the red color of these red blood cells. That's the hemoglobin protein. And then keratin protein, of course, would be found in this rhinoceros's horn. Let's go through a few more examples of important proteins. An antibody is a protein that fights infections by tagging bacteria and viruses for destruction. So you have special white blood cells called plasma cells, and they secrete these antibody proteins which then attach to the foreign invader, the virus or the bacteria that's making you sick. And that tells other cells in your immune system to go and destroy that tagged invader. Anything that has an antibody attached to it will be destroyed by your immune system. So this is part of how we fight off infections. Notice that not all hormones are proteins, but many of them are. Hormones are chemical signals which travel through the bloodstream from one part of the body to another. Now, some hormones are steroids, like testosterone and estrogen, but many hormones are proteins, such as insulin and glucagon that we've talked about before. All of the cells of your body are covered in what are called cells. Well, they're not covered, but they have on their surface cell surface proteins, which help control the movement of molecules into and out of cells. And some cell surface proteins they basically function like little flags, little markers that tell your immune system that, hey, I belong in this body, don't kill me. Because you don't want your immune system to kill your own healthy cells. You only want your immune system to kill foreign invading cells like bacteria. So cell surface proteins can also help your immune system identify which cells belong in the body and which ones are invaders. And finally, enzymes. There's an entire class of proteins called enzymes. These are proteins that speed up the rate of chemical reactions in our cells. So there might be a chemical reaction that would happen on its own, but it would take days, weeks, months, years for that chemical reaction to occur. Enzymes allow that chemical reaction to happen in seconds, milliseconds even. So their job is to dramatically speed up the rate of chemical reactions within cells. So let's take a closer look at how enzymes function. So in this series of images, the purple blob that you see is an enzyme. The name of the enzyme is sucrase. And every enzyme has a very specific three-dimensional shape. And specifically, there's going to be an area on the enzyme that has an exact perfect shape for some other molecule to bind to. That empty space with the exactly perfect shape is called the active site. The molecule that binds to the active site is called the substrate. So notice how perfectly that substrate fits into the active site of this sucrase enzyme. Now, the reason it's a perfect fit is because the substrate in this particular case is sucrose. This is a glucose that has been bonded to a fructose. So this is a disaccharide. Remember, sucrose is just a molecule of regular table sugar. And so when you eat something that contains sucrose, this enzyme is waiting in your stomach, in your intestines, and the shape of the sucrose is going to fit perfectly into the active site. And the enzyme's job is to hold that substrate and hold it in just such a way that the chemical reaction can happen. In this case, water is going to be added to break this bond. Now, do you remember what that's called? The chemical reaction where water is added to break a chemical bond? That's right. It's called hydrolysis. It's the opposite of dehydration synthesis. When we break this bond, these two sugars are no longer connected. It is no longer sucrose. It is now glucose and fructose, and they are two separate monosaccharides, and they are now going to be released as products. Now the active site is empty once again, and it's ready to take in the next sucrose and break it down, take in another sucrose, break it down, always releasing the products afterwards. Now, I don't want you to get the wrong idea. I don't want you to think that the only thing enzymes do is to break things apart. Sometimes it's the exact opposite. Sometimes you'll have an enzyme that can bind with two substrates at the same time and join them together. So some enzymes build and other enzymes tear things apart. In other words, some enzymes carry out dehydration synthesis, bonding things together. Other enzymes carry out hydrolysis, which means using water to split things apart. So what we just went through in this picture is called, listen to this now, this is the fancy name. It's called an enzyme catalyzed chemical reaction. So this would be something that normally would take place inside a living cell. It's a chemical reaction that would normally be very, 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 very slow, but it happens almost instantaneously because this enzyme is present. The enzyme sucrase is allowing the substrate sucrose to very quickly be broken down. Now, there are a number of factors that can speed up this process even more, and there are factors that can slow it down. So let's talk about some of these factors that affect the rate or speed of enzyme catalyzed reactions. So the first one would be, and these are listed in no particular order, but just simply the concentration of enzyme. In other words, if you have a large amount of substrate that you need to have broken down, let's say this is sucrose and there's a whole bunch of sucrose and you want to break it down quickly, the more enzymes you have, the faster that's going to happen. The greater the enzyme concentration, the faster the chemical reaction. Likewise, the greater the amount of substrate, the more likely it is that the substrate and the active site are going to find each other and bind, so that too can speed up the chemical reaction dramatically. So when we talk about factors affecting the rate of enzyme catalyzed reactions, an increase in enzyme concentration will always speed up the chemical reaction. An increase in the substrate concentration will also increase the reaction rate to a point. Now you get to a point where it doesn't matter how much more substrate you add, the enzyme is sort of maxed out and it's already breaking down the substrate as fast as it can. So this one is not as big of an impact as the first one. But probably one of the biggest factors that affects the rate of these chemical reactions is the availability, now there's a strange word, the availability of the active site. So let's go back and look at the picture. So under normal circumstances, the shape of the substrate will fit perfectly into the shape of the active site. And when those two fit together, the chemical reaction will take place. This is sometimes called lock and key theory, because the shape of that substrate perfectly matches the shape of this active site, just like the shape of your key perfectly matches the inside of your lock on your front door at your home. But if you tried to take your house key and use it on my classroom door, the shape won't match. It won't fit, and you will not be able to operate the lock. Every enzyme has a very specific shape on the active site that will only match up with one type of substrate. And anything that interferes with these two things binding, if you interfere with those two coming together and binding, you're going to slow down the chemical reaction. Now, one way this happens is excessive heat. If an enzyme gets too hot, all the little hydrogen bonds that are holding the shape of the enzyme, they start to break. And the enzyme will start to unfold. Remember, enzymes have tertiary structure, right? They're proteins. They have tertiary structure. If you heat it up, it's going to unfold partially. And look at what that does to the shape of the active site. It totally changes it. We call this a denatured enzyme. The active site no longer has the correct shape. It can no longer bind with the substrate. And so the chemical reaction is just not going to happen. It's going to dramatically slow down the rate of the chemical reaction. Another thing that will cause that same issue is drastic change in pH. So instead of heat, if you expose this enzyme to a really strong acid or a really strong base, that will also interfere with the hydrogen bonds in the middle of the folded up enzyme, and it'll change the shape, denature the enzyme, and the substrate will not be able to bind. So when we talk about affecting the availability of the active site, the obvious way that can happen is if heat or a drastic change in pH literally changes the three-dimensional shape of the active site. Obviously, that's going to slow down the chemical reaction. Now, there's a couple other factors that sometimes come into play. So if you notice this enzyme, here's its active site. Notice the shape. Here's the substrate, which fits perfectly like a lock into a key. But suppose there's some other molecule floating around inside the cell that has almost the same shape as the substrate. Not exactly the same, but it's close enough that it can slip in here and sort of physically block the substrate from binding. We would say this molecule is a competitor. It's a competitive inhibitor because it is competing against the substrate to fit into this active site. So if you have a large amount of competitive inhibitor floating around inside the cell, that's going to slow down the chemical reaction because it's preventing the substrate from binding like it normally would. So let me back up. So that's another way the active site can be made unavailable. It can be physically blocked by a competitive inhibitor. And then the final way that this can happen, oftentimes, not always, but oftentimes the enzyme will have not only an active site, but it'll have a secondary location, which is sometimes called the allosteric site. And sometimes you'll have a chemical, some sort of chemical compound in the cell that does not have the same shape as the substrate like we had in this example, but it'll have just the right shape to plug in to that backdoor site, what we call the allosteric site. And notice what happens. When this molecule is plugged in, it changes the shape. It alters the shape of the active site. So even though there's nothing physically blocking it, the shape has changed, and therefore the substrate cannot bind. So if you want to slow down an enzyme-controlled chemical reaction, you can heat up the enzyme or subject it to a strong acid or a strong base, and that will change the shape of the whole enzyme. And that will block the substrate from binding to the active site. Or you can introduce a competitive inhibitor, which will physically block the active site so that the substrate cannot bind. Or the third way, a non-competitive inhibitor can bind to this site, the allosteric site, changing the shape of the active site. So these are all the factors that affect the rate of enzyme catalyzed reactions. I want to finish with this really cool graph. Sometimes on the biology end of course test or the AP exam, you'll be shown a graph like this, and you have to be able to interpret what's going on. Just imagine if you had this graph with all the numbers and everything, but suppose it didn't have these written descriptions right here. You would have to figure out what's going on just based on the graph. So notice this is apparently a human enzyme, and we're measuring how fast the enzyme is doing the chemical reaction at different temperatures. So at really cold temperatures, the chemical reaction is really slow. As the temperature warms up, 10 degrees Celsius, 20 degrees Celsius, 30 degrees Celsius, notice the rate of the reaction speeds up. It gets faster. And that's because heat energy causes molecules to move faster. It increases the kinetic energy of the molecules, and they bump into each other. The substrates and the active sites start bumping into each other a lot more often just because they're moving faster. And so the chemical reaction gets faster, 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 faster. Now, the peak temperature for this particular enzyme is 37 degrees Celsius. That's the same as 98.6 Fahrenheit. That's normal human body temperature. So this is apparently a human enzyme that's doing its job somewhere inside your body. If you have a fever, like a really bad fever, and your temperature gets above 37 Celsius, like 40 or 45, notice your chemical reactions are going to slow down, because remember what happens to enzymes when they get too hot? They denature. The hydrogen bonds inside the enzyme break. The enzyme unfolds, it loses its tertiary structure, and it becomes more difficult for the substrate to bind to the active site. At extremely high temperatures, notice the chemical reaction pretty much comes to a halt. So one of the factors, remember, that can dramatically change the rate of a chemical reaction is to denature the enzyme that controls that reaction. This is the conclusion of our lesson on proteins. I hope you've enjoyed it. I hope you've learned something, and I will see you soon. Have a great day.